So it's given me enormous pleasure to introduce Doug Wallace today, and I've had a quick view of his slides. You're going to find this fascinating. So Doug is the director of the Center for Mitochondrial and Epigenomic Medicine at the Children's Hospital of Philadelphia, CHOP. He holds the Michael and Charles Barnett Endowed Chair in Pediatric Mitochondrial Medicine and Metabolic Disease, and is also professor of the Division of Human Genetics, Department of Pediatrics, Perelman School of Medicine, University of Pennsylvania. Now, more than 35 years ago, Doug and his colleagues founded the field of human mitochondrial genetics. The mitochondria, as you know, are the cellular power plants, organelles that generate most of the cell's energy. Mitochondria also contain their own DNA, the mitochondrial DNA, which encodes the wiring diagram for the cell's power plants. Doug showed that the mitochondrial DNA is inherited exclusively from the mother, and the genetic alterations in DNA can result in a wide range of metabolic and degenerative diseases, as well as being important in cancer and aging. And one of his seminal contributions has been the use of mitochondrial DNA variation to reconstruct the origin and ancient migrations of women. So it gives me great pleasure to introduce Doug. Following Doug, Betony will give a talk, and I'll give a small introduction to Betony because she'll summarize the talks of this session. Doug, if I can ask you to present your talk and paper, it's over to you.
So the question we have today is there some relationship between mitochondrial biology and its relationship, and is there a relationship with quantum phenomena? And of course, this is all part of this spring's discussion. That's fall, right? Fall's discussion on the quantum mitochondria, energy, information, and implications of health and disease. And as I said, Larry Grossman, Alish, and Shexing, and also Peter Burke have been very involved in my conversations over the years, and more recently, Matthew Fisher. So I'll mention people as we go along. But the way we got involved in this was a very simple concept, and that is to be alive is not just to have structure or anatomy, but you need to be animated. And to be animated, it requires energy flow. You also need information to maintain the anatomy, but you also need information for energy flow. And what has been really problematic, I believe, in Western medical philosophy for the last basically half a millennium is that it's been based primarily on an anatomical perspective. And that goes back to Vesalius, who was the first to define and publish a pretty accurate analysis of the human body. And that was so great that, of course, everybody wanted to study the human body in the same way. But what they missed is the basic idea that it's okay to study anatomy if you're a medical student and you dissect an anatomy and learn a lot about anatomy. But the problem with the cadaver is it's dead. And so the real question is, what's the difference between live and dead? And it seems to me that's the most important question, and one that gets very little attention in medical school, at least the ones that I've taught at. So we decided more than almost 50 years ago now to decide to look at the role of energy in human health and disease. The reason that we decided to work on mitochondria is because mitochondria makes most of the energy of our body, and it does so by burning hydrogen in our food with the oxygen that we breathe, and it generates an electrical capacitor, and that capacitor then is a very physical phenomena, and therefore brings us closely to the physical sciences. And what's interesting about biology as far as animals are concerned is that we're at the top of the food chain. So as at the top of the food chain, energy becomes limiting. Therefore, it follows logically that evolution would create the most energy efficient system it possibly could, because there would be tremendous advantage to do that. So therefore, we can imagine that energy production would take advantage of every possible way of maximizing energy output, and that could include application of quantum phenomena. So the idea then is that we should look for quantum phenomena in biology, but then the question is, where would we look? And we propose to look at the brain. And the reason is that many cells in the brain, specifically the parvalbumin interneurons are at their maximum energy capacity. So they are basically using all the energy that the mitochondria can generate to fire at their very high depolarization rate. Therefore, we would imagine that if there's going to be a quantum effect or quantum biology for the mitochondria, it's going to be where the maximum energy demand is, and that would be in the brain. And as I know that you know, that almost 20% of the total mitochondrial body energy goes to running the brain. So we then decided to look at this phenomenon many years ago, and we took a number of different approaches. One is genetic, which is, I started out as a geneticist, and then biochemical, and then more recently physical, with the interaction closely with Clarisse and Peter Burke. So, let's see. So just to refresh your knowledge, your cell is made of two life forms. One life form forms the nuclear cytoplasm. That was originally an archaeobacteria. And the other, shown in as a circle here, was an aerobic bacterium that entered our cells about 2.5 billion years ago. And these two systems maintain parallel information storage and retrieval systems. Each has its own DNA that's transcribed into RNA, translated onto proteins to make structure. And the mitochondrial DNA is about 16,569 base pairs, codes for transfer RNAs, ribosomal RNAs, and 13 polypeptides. And those 13 polypeptides are translated on mitochondrial ribosomes, and they go to make the essential enzymes to make energy by oxidative phosphorylation. So it makes 7 of the 45 proteins of what we call respiratory complex 1 of the 11 proteins of complex 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. So as Jeffrey says, we can envision the mitochondrial DNA encoded proteins as sort of the wiring diagram for the power plant. Where the power plant has all the structural genes for the power plant, about 1,500 in the nucleus, to assemble the system. But to run the energy generating system, oxidative phosphorylation, it requires key proteins that are made in the mitochondrial DNA. And as I said, we basically burn hydrogen with oxygen. So if we think about glucose, goes through glycolysis to pyruvate, pyruvate then goes through a pyruvate transporter and pyruvate dehydrogenase to drive the TCA cycle. And the purpose of that is to strip the hydrogens off the hydrocarbons to give you NADH. And then the hydrogen is burned by complex 1, CoQ, complex 3, cytochrome C, complex 4, where the electrons then react with oxygen to give you water. And as the electrons flow down this electron transport chain, they pump protons across the inner membrane, which is an insulator, and you give positive and acid on the outside and alkaline and negative on the inside. And Peter Mitchell won the Nobel Prize for this idea that mitochondria is basically a physical capacitor rather than substrate phosphorylation. And then the membrane potential can be used by the ATP synthase to condense ADP and phosphate to make ATP, and then it's exchanged across the inner membrane by the adenine nucleotide translocator, where it can do work. And that is an oxidation with phosphorylation, and hence oxidative phosphorylation. And electrons from the beginning of the electron transport chain are electronegative enough, they can be put onto O2 to give you an unpaired electron, superoxide anion. Manganese SOD will take that to hydrogen peroxide, and then this can be reduced by NADPH and glutathione peroxidase to water, but this is rate limiting step. So we build up hydrogen peroxide in the presence of a transition metal. We'll get another electron to give you hydroxyl radical, and these are the reactive oxygen species that damage proteins, lipids, and DNA. And the mitochondria has a number of other important features, self-destruct system called apoptosis. So basically then the mitochondria regulates energy, redox state, generates reactive oxygen species, regulates calcium by uptake using the electrochemical gradient, regulates cell death, and these intermediates are critical in regulating the epigenome. So the mitochondrial DNA, which we're going to focus on today, for simplicity is, as I said, a circular molecule. It's for a small and large ribosomal RNA, 22 transfer RNAs that punctuate all of the key protein genes, and then it codes for 7, ND1, 2, 3, 4L, 4, 5, and 6 for complex 1, cytochrome P for complex 3, CO1, 2, and 3 for complex 4, and ATPase 6 and 8 for complex 5. Now, the mitochondrial DNA, because it's a wiring diagram, and it turns out has a very high mutation rate, then almost everybody has slightly different mitochondrial DNA genotype. And since this system requires that all of these enzymes, these 4 enzymes, use exactly the same substrate for the same purpose, then they all must be electrically balanced to manage this electrochemical gradient appropriately. Therefore, it follows that these must co-evolve so that you don't have a situation where one of these becomes less efficient for maintaining the proton gradient and would short circuit the system. So the way that is done is to have this mitochondrial DNA inherited only from the mother, and then every new mutation is tested against the previous variation. And if it's good, it's retained. But if it's bad, that is, if it's out of sync with the wiring diagram, then it becomes deleterious, and we call that mitochondrial disease. And there are hundreds of thousands of mitochondrial DNAs in each cell, and they're replicating, giving mutations, and that causes cells to have a mix of mutant and normal mitochondrial DNAs called heteroplasmy. And as the cells undergo division, let's say this is an oocyte, it can give rise to cells with different percentages of mutant and normal. And as the percentage of mutants increases, the energy output of the cell declines until it falls below the minimum for that organ to function. And then you get malfunction, and we would then call that unhealthy or disease. And of course, since the brain is using much of the energy, that's going to be one of the first organs to be affected. Now, because the mitochondrial DNA can only change by sequential mutations, and if you sequence the mitochondrial DNA of any 2 individuals, then the number of nucleotide differences is proportional to the time they shared a common mother. So what we did is just sequence the mitochondrial DNAs from indigenous people around the world, and we could reconstruct then the migration of women. With origin in Africa about 150,000 years ago, radiation into a series of different types. For this is the Khoisan of the Kalahari. These are 2 different pygmies. These are sub-Saharan Africans. And then out of Africa, only 2 mitochondrial DNA successfully left and colonized all the rest of the world. M moved all the way down along the tropics into Australia, and then much later moved into the temperate zone. But N moved directly into the temperate zone, giving rise to a whole series of European type lineages that have these letters H, J, T, U, U, K, V, and then migrated laterally to intermix with the M lineages in Asia. And then ultimately, only a few mitochondrial DNAs across the Bering Land Bridge colonized the Americas. Our idea is that as the mitochondria moved into different environments, mitochondrial mutations arose that allowed people to adapt to those environments by changing their energy metabolism. And that's why we have regional specific mitochondrial lineages, because they're adapted to local environments. And so basically the mitochondrial DNA has 3 types of mutations: ancient adaptive polymorphisms, which we just mentioned, recent deleterious mutations, which are disease causing, and then the accumulation of somatic mutations as we age, and that's the aging clock. But these are just a few examples of mitochondrial DNA mutations, like this mutation in the tRNA leucine gene at 3243. If it's 20% to 50% mutant, it will give you diabetes. 50 to 80% gives you neuromuscular disease, and at 100% kills you as an infant with Lee syndrome. Another mutation here at 11778, you inherit that from your mother, and in midlife you'll lose your vision and be legally blind, Leber's hereditary optic neuropathy. So there are almost 1,000 now putative pathogenic mitochondrial DNA mutations. But what we're going to talk about are these adaptive mutations that were involved in this migration. So this is a variant site that's found in about 3/4 of all sub-Saharan African lineages. Variant H is found in half of Europeans. Variant A, B, C, and D, they arose in Asia, across the Bering Land Bridge, and colonized the Americas. And you will see that these variant lineages predispose to specific kinds of diseases. So now to talk about mitochondrial physiology. So what we discovered, and this was Martin Picard and Megan McManus and our colleagues at Jefferson, we were looking at mitochondria in the heart, and we found this how amazingly compact the mitochondria are. And when you do a tomogram of the mitochondria, what immediately strikes you is that all of the mitochondria cristae, these are enfoldings of the mitochondrial membrane, are aligned with each other. And that was quite shocking because that implied that these cristae must be somehow interacting with these cristae across what seemed to be an impervious barrier. So the question is then, how could these cristae be talking to these cristae? And basically, the mitochondria then, remember, is the electron transport chain, pumps protons. So if you open up this cristae, this being a cristae, and we'll call this the cristae lumen, if you open this up like Peter Mitchell's diagram, all the protons would be out here and all the hydroxyl ions would be here. But what we found is that the cristae structures, which are these very small lumens along here, are in fact closed by OPA1 and the Mekios complex. So that means that all of the protons are concentrated in this very, very tiny volume. So we have then a very high electrostatic field. And we wanted to know whether that we could prove that. So what we did is, this is beautiful work from Peter Burke, is we took mitochondria, we linked them to antibodies and then bound it to a graphene film. And the graphene film will cause a flow of electrons to the pi orbitals, but it's very pH sensitive. So you can put an electrical potential across this and then measure the amount of protons that are leaking from the mitochondria. So what you can do to a mitochondria is you can put in what are called uncouplers, which basically punch holes in the inner membrane and release the charge. And so when you do that, you would expect the hydroxyl ions in the matrix to go out and for the pH to go up as the membrane potential collapsed. And that's exactly what you see. But then what we did is we took advantage of the fact that OPA1 is cleaved by PIM, which is a pro-apoptotic protein. So we could then just treat the mitochondria with PIM, which cleaved the Mekios complex. And if we were right, the proton should be released. And instead of going alkaline, it should go acid. And that's exactly what we found. So basically then all of the protons are pumped into this very, very tall, small volume. And that means that the electrostatic potential is much greater than Peter Mitchell ever imagined, with the protons distributed across this outer portion between the inner and outer membrane. So if you then stain a mitochondria, and this is very nice work from Peter, you stain mitochondria with a dye that's charge related, you can see that all the dye is primarily bound to the cristae, whereas the mitochondrial DNAs, which stain here in green, are in the matrix. So this then implies that, and in fact, there's a very strong charge differential between the matrix and the cristae. So how does this happen? As we said, when you burn NADH to NAD, the electrons flow down through complex 1, complex 3, complex 4, and they pump protons. So that gives you then this high membrane potential of protons in the cristae lumen. You can imagine if you kept pumping protons, ultimately the electrostatic potential would be so great that it would destroy the membrane. So there must be a rectifier. And what we discovered is that, in fact, the adenine nucleotide translocator, which people originally had only thought was involved in ATP-ADP exchange, is also a voltage-sensitive proton channel. So if we do a patch clamp of a mitochondrial inner membrane, and we now change the voltage across the inner membrane, we can see that the amount of proton leak increases as the voltage goes up. If you use a tractyloside, which binds to the translocator, you can completely obviate that. And if you use one of our mice with a mutant in the translocator, it completely knocks out this effect. So basically then, the mitochondrial adenine nucleotide translocator is a major rectifier of the voltage potential. And so that gives you the potential of pumping protons. And then when you get to a maximum membrane potential, opening the rectifier and having the protons go back. So now you have the potential of an oscillating circuit. So this then suggested that maybe there would be a way for some signal transduction to occur by these oscillations in the membrane potential. So what Peter designed was to create then electrodes, and then he used carbon nanotubes, which are can conduct electricity based on the potential around them. So we made a tiny little tube that we could flow the mitochondria down across the carbon nanotubes. So that's what this would look like. And then we can measure the oscillation of the membrane potential as the mitochondria cross the carbon nanotubes. So this is the without any mitochondria. Then a mitochondria comes in and then it goes out, and it comes in and goes out. But what struck us was this oscillation here. So this then could be amplified to show that as the mitochondria sat on the carbon nanotubes, it itself was oscillating. So that implied that maybe there was an oscillating potential in this system. So what was done then is to make a whole series of these small 1 micron by 5 micron channels, feed the mitochondria, isolated mitochondria in there till they stop, and then infuse in substrate. So what you would expect then is if you had a potentiometric dye, say TPP, that's fluorescent, and it then would be taken up as the membrane potential increases, and it would then be released as the membrane potential declined. So we hypothesized that we might see an oscillation in the uptake and loss of this potentiometric dye. So this is looking at the mitochondria trapped in the tubes, and you can see that as the substrates come in, the mitochondrial membrane potential lights up, takes up the dye, and then it starts oscillating. And this suggested that all of the mitochondria were indeed oscillating by some kind of physical phenomenon. So that then raised the possibility that maybe the mitochondria was in some way generating a field. Now, we don't know what that field is. We don't know what its characteristics are. It's a hypothesis. But if a mitochondrial field is there, it might explain why the membrane, why the cristae of the mitochondria were aligned, because they have this very high differential in charge. And that would then cause them to repel each other. So if there were such a field, and we would like to find such a field if there is one, then what would it interact with besides other mitochondria? Well, you can imagine if one mitochondria is oscillating at a particular frequency, then the adjacent mitochondria would have to oscillate at the same frequency, because if not, they would be out of phase and they would compete with other and negate the energy outflow. But if they're oscillating in phase, then in fact, maybe that would amplify the effect of the oscillation and allow it to be propagated across the cell and maybe even between cells. So if that speculation is right, and I reiterate that that's a speculation, then what might be the target? Now, this is not our work, but I think it's very interesting ideas. So Matthew Fisher has proposed something called Posners might be an important way of storing information, and that turns out to be in the mitochondria. So Posners have phosphates that are interacting with calcium, 3 calciums. And the Posner has a nuclear spin of 1/2. Why would Posners be relevant to the mitochondria? Because as I told you, the mitochondria takes up calcium, but it also takes up phosphate because it has to make ATP. So therefore, the mitochondria naturally generates calcium phosphate crystals. And calcium phosphate crystals, according to Matthew's studies, ultimately generate what look like Posner molecules. And his idea then is that these phosphates with a nuclear spin of 1/2 could interact with each other. And in fact, if you had Posners interacting with each other, these phosphates could actually become entangled with their nuclear spin. If that's the case, then something has to, in fact, decide what would be the entanglement of the mitochondria of one cristae, one mitochondria versus another. And that's why maybe some kind of field effect might be important. Another idea of interest, and this is from the Usselman paper, is that in fact, the riboflavin, which is a key electron carrier in respiratory complex 1, 2, and 3, that riboflavin might actually have a semiquinone superoxide spin radical pair situation. So the idea that is that an electron comes in to the system, and then it can either the exit or another electron can come in, and depending on the state of the system, you can either get a singlet or a triplet state. And they were using what's called Namor frequency oscillation in the Earth's magnetic field, about 50 microtorr, and they can cause the mitochondrial, the spin of the electrons to become mixed, and that can either give you a singlet state with anti-parallel spins or a triplet state with parallel spins. And they argue that then can be used to modulate mitochondrial ROS production. And this is actually data that they have. So they're using then this Zeeman magnetic field to scramble the electrons in the presence of the Earth's magnetic field. And what they find is, depending on how they position the field relative to the mitochondria, they can get increasing mitochondrial reactive oxygen species. And what was surprising about this paper is, depending on the either perpendicular or parallel direction of the field, you can either change mitochondrial electron transport chain, that is, this is a measure of oxygen consumption, or they could actually change glycolysis, that is, by seahorse, the so-called ECAR analysis. So their idea is that maybe the flux rate of electrons through things like the flavines can generate then either superoxide anion on one side with a triplet state, or hydrogen peroxide on the other with a singlet state, and that they envision then is interacting with the mitochondria to generate a physiological signal. So the idea then is, and again, at this point, all speculation, the mitochondria may be then generating some kind of signal. It then may have antennas, which could either be Prosners or could be these radical pairs. And they then in turn can be transposed into a biochemical output, which is through the mitochondria. Now, that of course is what this discussion is going to be all about. But what can we do that says that there might be something interesting in the brain that relates to mitochondrial oscillations? So here is our model. This is one mitochondria. This is another mitochondria. These are the cristae that are aligned with the high level of proton charge. And we're arguing that the adenine nucleotide translocator is rectifying the proton pumping and giving you some kind of oscillation, and also driving the ATP synthase to make ATP, but that's an aside. Oh, by the way, it's very interesting though, ATP synthase is a dimer, and if you look at the cristae, all the dimers are on the outsides where the charges would be at the highest potential. Okay, so given that idea, then what do we have in relation to the brain that might relate to this idea? Well, first of all, the membrane potential of the mitochondria is about 180 millivolts, whereas the membrane potential of a neuron is anywhere between 30 or 90 millivolts, or more likely about 50. There are 1,000 mitochondria, 10,000 mitochondria in a neuron, but there's only one outer membrane potential. So the idea is that the amount of charge and charge oscillation for the mitochondria is far greater than the amount of charge oscillation for the action potential of a neuron. And maybe, in fact, things like the EEG is not about axon potential migration, but is about oscillating energetics of the mitochondria. Okay, so given that speculation, this is the work of my colleague, Tim Roberts. And here he's put a patient, a child, into a detector of magnetic fields. And he can then ask the child to do mental activity and shift the magnetic field. But the point I want to make here is this is a false color of the relationship between frequency and time based on the challenge. And you get this very unique pattern of a normal person, which if you look at autism patients, it's completely gone. So that means that something about autism has knocked out this frequency of the EEG, which he's sensing by a magnetic field. Okay, So that would then say there's something in the brain EEG oscillation that is related to autism. And then the question is, what is autism related to? So what we did is we looked at mitochondrial DNA lineages, haplogroups, relative to risk of autism. So these are European mitochondrial DNA lineages, and the odds ratios for getting autism, if you have one of the European lineages, IJKTU, relative to the common one, H, is up to about 2, odds ratio of 2. The odds ratio of getting autism if you're a male is 4. So these mitochondrial lineages are half as important in risk for autism as the difference between male and female. And these are highly statistically significant with multi-testing correction. So these particular mitochondrial DNAs represent about half of all European mitochondrial DNAs, therefore implying that half of European males have a uniquely high predilection to autism. But you can say, okay, that's all very interesting, but that doesn't connect oscillations in the brain with mitochondrial DNA. So what we did is we made a mouse in which we imported a particular known human mitochondrial DNA mutation. This is in the ND6 gene, changes proline at codon 25 to leucine. So we took cultured mouse cells, we mutagenized them, we screened them for mitochondrial respiratory deficiency. We then sequenced all the mitochondrial DNAs, and after many, many months, years, we found exactly the same mutation that changes proline at codon 25 to leucine in the mitochondrial DNA. We then took this cell, removed its nucleus by enucleation. We had made a female pluripotent embryonic stem cell. We removed its mitochondria with a drug. We fused the mitochondria from the mutant cell with the pluripotent nucleus to give you a mitochondrial cybrid with the pluripotent nucleus, put it in a blastocyst into a foster mother, got the chimeric females and bred them and picked up the mitochondrial DNA mutation. So now we have a mouse with the exact same mutation as we see in a patient. Okay, so this mutation then is going to affect respiratory complex 1, and that's going to decrease the rate that NADH is turned into NAD. So what we see, if we assay respiratory complex 1, we get about a 50 to 60% reduction in specific activity. And if we use then what's called fluorescence lifetime imaging microscopy and look at the hippocampus of the brain, we can see there's far more NADH in the mutant mouse brain than the control mouse brain. And what that does is greatly increase the mitochondrial reactive oxygen species production and result in swelling of the axons and their slow degeneration. Okay, so we then made a mouse with a point mutation that we've established gives you a partial defect in mitochondrial function. We made another mouse with a mutation in the ND5 gene at serine 204 proline. And then we do the mitochondrial respiration studies. This is the normal mitochondrial DNA. This is the ND5 mutant, and this is the ND6 mutation. So you can see that there's a perfect correlation between the different mutations and the effect on respiration, which is quite subtle. Okay, so then we put the animals into a neurobehavioral test. So this is called the zero maze, and the mouse is put here, and then it has to decide if it wants to venture out and find out what's on the other side, and being concerned that it might fall off. So if we look at the ND6 and the ND5, I mean, the control ND5 and ND6, we see that there's exactly the same decline as we see in the respiratory decline. And we can then look at, this is a depression assay, the amount of time that the mouse is willing to swim. We see then that the ND5 and ND6 are more prone to depression, but when we give them lithium, it completely wipes out that effect and they just swim until we finally take them out. And finally, if we ask the ND6 mouse to have a social preference in the social preference study, it would prefer to use and interact with an inanimate object rather than an unknown mouse. It's hypercompulsive by marble marrying, but here's the bottom line. It has the same electroencephalographic change as we see in autism patients. So the point I'm trying to make here is we made a point mutation in the mitochondrial DNA, changed energetics subtly, got all the phenotypes of autism, and the same electroencephalographic change that we see in patients. Therefore, mitochondrial physiology is regulating the EEG. That's the point I want to make. So the EEG is an amazing thing because it oscillates across the entire brain. It's not about one neuron or about 100 neurons, and neurons are all in different directions. So how do you get a coherent oscillation of field? And I'm hypothesizing that maybe mitochondria generates a large enough field that you can get oscillation that is coherent across tissues and maybe even organs. And that in a mild defect in the brain, in energetics, will destroy the ability to maintain those coherent fields and will give you neuropsychiatric disorders, or to wit, maybe all neuropsychiatric disorders are bioenergetic. Okay, so with this idea and a lot of other studies, we have been arguing that if you take anatomy out of the middle of medicine and you put energy, and all the common diseases have the same pathophysiological mechanism. They can have a defect in energetics that'll increase ROS, reduce the redox potential, increase calcium. This will cause damage to the mitochondrial DNA polymerase, which then makes errors in replication that accumulates mitochondrial DNA damage, resulting in progressive decline. And that we believe is aging, and why diseases can have a delayed onset and progressive course, because this accumulated defect and augments a subclinical pre-inherited defect in the body. So you can have alterations in nuclear genes or changes in the epigenome. You can have ancient adaptive mutations or recent deleterious mutations. You can change the source of your carbohydrates and fats. You can change whether you exercise or not, or you go on caloric restriction, or you can smoke and poison your mitochondria. If you have a partial defect in mitochondria, and remember, in these patients, these cell lines, this is only about a 20% reduction in respiration rate. If you have a partial respiration rate, you get neuropsychiatric disorders. A little bit more, you'll knock out, affect the heart, the muscle, and the renal system. And these are all the organs with the common diseases, the high energy organs. If you inhibit mitochondrial function, you'll build up fatty acids and carbohydrates, and that's what we call metabolic syndrome, and that will give you obesity. Mitochondria is, in fact, a bacterium, and it's now clear that the mitochondrial DNA and mitochondrial peptide components are major signaling systems for the innate immune system. So the mitochondria then regulates the innate immune system, and that's why energetic deficiencies give you common diseases that also have inflammation. And finally, of course, as you heard from Afshin, environmental factors such as AIDS be inhibiting mitochondrial function. And we're quite, as a team, the COVID team, quite interested in whether the long-term effects might be long COVID. And of course, cancer is all about manipulating energy. Okay, so that's basically the idea, but where would you go if you want to speculate any farther? We had, until COVID-19, an institute in Xi'an, China. And our idea in Xi'an is that for the last 5,000 years, physicians, we'll call them, in China have been experimenting with different things to see what make people feel better. And they found a whole bunch of herb combinations and so on. And they call this idea of sort of human energy what we call qi. And this is the symbol of our center here at Center of Mitochondrial and Epigenomic Medicine. So the problem is that Western medicine says, you know, what is qi? It doesn't make any sense. It doesn't have a physical component, and we all believe in anatomy. Well, I'm saying, yes, Western medicine is about anatomy. The mitochondria is the anatomical conduit for energy. And therefore, the mitochondria then links Western and Eastern medicine together. And that this is the common factor that has been explored in most of traditional medicine. And that then leads to some rather interesting speculations. Well, then phytochemicals might be very interesting, and we're already exploring phytochemicals as new therapeutics. But what about acupuncture? Acupuncture is all about energy, what do they call it? I forget the word, energy pathways. Well, what if, in fact, in a region of the body, you have impaired mitochondrial function, the mitochondrial oscillation begins to become aberrant, as we think is happening in autism, and then that disrupts the this pathway of energy, and then gives you the essential feeling of unhealthiness. So if you then modulate the mitochondrial oscillation at that point, you can reconnect the pathway and thus generate a healthy chi. And that means that, in fact, what are Tai Chi and meditation and all of these things doing? Well, maybe they are also using neuropsychiatric interactions to modulate mitochondrial energetics and to maintain this oscillating system that we are arguing might be the central feature of health. So with all that madness, these are the people that are probably embarrassed to be even embarrassed associated with me. But here's Peter Burke, which I've worked with a very long time and has done much of the physics that we've talked about. And more recently, we've been working closely with Clarisse, and that's been a real joy. Katie, Jessica, and Orion have been very important in studying the mitochondrial ND6 point mutation and ND5. Tal, Anna, and Eric did all the work on the mitochondrial mutants autistic phenotype. And then this is Tim Roberts, who, of course, specializes in the magnetoencephalographs. And then these are all the people that make the system work and are able to help us do this work. And I do want to recognize both IONUS and NIH for supporting this work. And that's the story, and you're welcome to it.
Doug, thank you very much indeed. Absolutely fascinating. I'm sure there are going to be a lot of questions in the discussion there, and a number of themes that we've been pursuing for many years, and you've laid them out beautifully in your presentation today. And so we're looking forward to the discussion. Can I now introduce Betony, who needs actually no introduction. Betony works with us and looks after our scientific communications. She's undertaking a PhD in quantum biology at the University of KwaZulu-Natal. I think she has a master's in physics and a master's in English literature as well. And she's going to give us a brief recap of the talks throughout this autumn session, these autumn sessions, which would then lead us nicely into the roundtable discussion. So Betony, if I can ask you to present your paper.
Hello, everyone. As Jeffrey mentioned, as a warm-up for the discussion, I'll do a quick recap of the various presentations. As you probably gathered, and following on from Doug's wonderful presentation, the Gaia Foundation has had a long-standing interest in the role that mitochondria play in biological systems. While many of the series have addressed topics associated with mitochondria, the autumn series focused explicitly on how mitochondria respond to different or extreme conditions, how they've optimized their function with respect to Earth's specific magnetic and gravitational fields, and the implications this might have for health and disease. There were many, many ideas that came up in this series, so forgive me if I've left out any important ones. The series started with a presentation from Prof. James Moon, who gave an overview of the COVID-Sortium project. At the onset of the SARS-CoV-2 pandemic, he repurposed a research team that was more familiar with cardiac imaging towards developing a strategic approach to clinical academic research in the time of the novel coronavirus. The project moved from idea stage to ethical approval at a very fast rate, with full permissions granted in 7 days. And the speed with which it enrolled meant that funding was not guaranteed, and when it did come, it came from atypical sources. The COVID-Sortium has been an academic success story, generating 20 papers so far, published in high-impact journals such as Science and Nature. But while the publication record of the project has been impressive, it has been coupled with a commitment not to compromise on scientific integrity. With the initial urgency of the pandemic response now abating, lessons learned from the COVID-Sortium approach might be applied to the more general case of academic research. While not explicitly about mitochondria, the COVID-Sortium project might offer valuable insights for an endeavor such as quantum biology, which by definition is interdisciplinary and demands collaboration. And the COVID-Sortium approach prioritizes a simple, deeply resourced, collaborative, and fully committed stance and atypical leadership, an approach which might be adopted to answer other big questions. So to complete session one, we had Prof. Alistair Nunn with his thoughts on COVID-19, long COVID, mitochondria, and quantum mechanics. Prof. Nunn addressed the various ways in which mitochondrial function, quantum biology, and COVID infection might be intertwined. The Gaia Foundation, as I mentioned, has a long history of interest in mitochondria and the role they may play in optimal health. And one risk factor for severe COVID may involve mitochondrial dysfunction incurred by less than optimal health, either due to genetic factors, age, lifestyle. In particular, there seems to be some sort of metabolic tipping point where the flexibility of mitochondrial function is stretched past its maximum healthy limit. Prof. Nunn traced this back to the origins of life and an ancient metabolic pathway called the Krebs cycle, which may have come into being in a thermal vent, where a flow of hydrogen into carbon dioxide created a strong energy gradient. These conditions, perhaps coupled to the existence of prebiotic cyclic chemicals and ion compounds, as well as the electric fields generated by the movement of ions, led to a self-organizing, far from equilibrium structure that obeyed the laws of thermodynamics, dissipating energy from the proton gradient and perhaps embracing quantum effects to function. In effect, the dissipative process generated compounds that form life, and in time, this life evolved to use some of these compounds to operate in reverse, effectively eating these compounds to generate its own proton gradients by extracting energy via the flow of electrons. Today, this process is largely undertaken in the mitochondrion, but coupled to a process called glycolysis that can operate without oxygen for a period to generate energy. However, the Krebs cycle can still run in the original direction to generate compounds used to build life. This process is also utilized during inflammation, which is why it is so tightly linked to hypoxia, as life originally evolved in a world with little or no oxygen. The SARS-CoV-2 virus can metabolically reprogram its host to help it avoid the immune system and generate metabolic precursors. The problem being is that this reprogramming is very similar to what happens in inflammation. And because the mitochondrion is central to most pathways, if the host's mitochondrial reserve is already compromised, it is possible that the virus can tip its host into chronic inflammatory cycle by overloading mitochondrial function. Prof. Nunn then suggested that the careful use of hormetic stimuli may be needed to restore optimal mitochondrial health and induce an epigenetic switch back to the modern homeostasis using hormesis following rest. And some of the ways in which this could be done might be the right dose of physical activity, calorie restriction, plant stress compounds, stimulation of regeneration of functional proteins, or using specific compounds to inhibit the inflammatory spiral. In the second session, the subject moved even more specifically to mitochondria under pressure, with Prof. Carl Morton speaking about mitochondrial dysfunction and the relevance for chronic fatigue syndrome and long COVID. One of the difficulties in treating chronic diseases is establishing cause and effect. And this is complicated by the fact that although there might be some clear cause of a disease such as long COVID, i.e. infection with COVID, there must be some other susceptibility factors as well as drivers that account for the fact that not everyone gets long COVID. This is similar to other post-viral syndromes, such as myalgic encephalomyelitis and chronic fatigue syndrome, which have puzzled doctors for years. Prof. Morton went into a lot of detail as to the challenges of defining and diagnosing chronic illnesses that I don't have time to address here. But in the interests of our theme, there seems to be growing evidence that mitochondrial dysfunction plays a role in post-viral syndromes such as CFS and ME. So one of the ways in which this dysfunction seems to manifest is in changes to the energy requirements of cells, as well as changes to electron transport chain proteins. And researchers are using metabolomics to investigate this by looking at what the plasma can tell us about what's happening in different organs. And preliminary evidence seems to indicate that there are really big differences in metabolites between CFS or ME patients and controls. But the problem is deciding which are the important metabolites, especially as there remain many unidentified metabolites. In long COVID, there's a lot less information, though there is some evidence that microclots might be causing some of the observed problems. Prof. Morton also addressed the possibility that mitochondria play different roles in the same cells. For example, they can act against pathogens, for example, persistent L-form bacteria. But if they are too preoccupied with this, they produce less energy, or if they are forced to produce energy, the pathogens can re-exert control, which would explain things like post-exertional malaise. One of the more interesting aspects of mitochondrial pathogen response that Prof. Morton touched on was the possibility that ATP might be produced outside of the mitochondria, which, as he acknowledged himself, would be viewed as treason by some. For example, high energy tissues such as the brain or plasmablasts have few mitochondria. So how do they satisfy their high energy requirements? Possibly producing ATP using other membrane structures. But if mitochondria are important in fighting pathogens, this lack of mitochondria also leaves them vulnerable to these pathogens. He also went into detail about the role that the microbiome, both the healthy microbiome as well as the pathogenic biome, plays in chronic disease, which I only have time to mention in passing. And finally, very quickly, he touched on new techniques, particularly Raman spectroscopy, with which to investigate some of these ideas. I mention this also because it is pertinent in the context of quantum biology, with its interest in vibronic coherence and electron transport chains. Raman spectroscopy looks at the vibrational spectrum of molecules. In effect, it's like a molecular fingerprint. And Prof. Morton's group has used this technique to look at, among other things, metabolism. I won't go into the details, but it might be a fruitful future avenue of research, bringing together quantum biology and the study of mitochondria. In session 3, our speakers looked at the role that magnetic fields play in biological systems that are related to mitochondria. Prof. Clarice Aiello gave us an introduction to quantum spin, which is the property that magnetic fields act on, and how spin may play a role in biology, and why it is important to develop high-tech experiments so that we might understand nature better by measuring it better. Why do we need such high-tech experiments? Well, because the things that we are measuring are incredibly small, and to measure incredibly small things, we need incredibly small sensors, i.e. quantum sensors. Prof. Aiello took us through the most commonly cited example of possible spin effects in a biological context, which is bird migration. It's very well accepted in spin chemistry, which was developed already in the 1960s, that magnetic fields can alter the final products of a class of chemical reactions. And these reactions depend on the spin state of the reactants in this reaction. And if these macroscopic spins can be changed by a magnetic field, then the macroscopic outcome of the reaction can be changed. Very importantly, this can happen at room temperature and at weak field strengths, such as the Earth's field. While this was developed in spin chemistry, it was introduced into quantum biology when Klaus Schottl hypothesized that coupled spins known as radical pairs could be responsible for avian navigation. It's been suggested that birds can sense the Earth's magnetic field effect on chemical reactions, and that this could act as a compass. Perhaps, and they're not sure exactly how it is transduced into a chemical signal, but perhaps by the translation of this magnetic field into retinal sensitivity, for example. It is generally accepted that this takes place in a protein called cryptochrome, which is present in a variety of organisms and is implicated in things other than navigation, such as circadian rhythms. So there is evidence for cryptochrome-based magnetosensitivity, but the experimental evidence occupies two very different scales. Radical pairs in cryptochrome consist of flavin and tryptophan-based radicals. On the nanoscopic scale, if you put cryptochrome in solution, it has been shown that flavin-based autofluorescence in cells is magnetic field sensitive. In particular, it is sensitive to weak magnetic fields, which is a hallmark of the radical pair mechanism. On the macroscopic scale, for whole organisms, the experiments are primarily behavioral. So bird migratory behavior is tested under conditions that may have an effect on the radical compass, like conditions or oscillating electromagnetic fields or cryptochrome deficient organisms. So far, however, there's been no completely unambiguous proof that it is spin at the heart of this mechanism. And there is the need to bridge these two scales, to directly link spin changes to biological function and to learn how to control these quantum degrees of freedom. Why should this matter? Because it turns out that radical pairs might be important in more contexts than just navigation. Magnetic fields might be able to modulate the balance of reactive oxygen species, which we know are important signaling molecules in biological systems that are also potentially damaging. And as Doug mentioned, mitochondria are one of the primary sources of ROS, and understanding and controlling of the effects of spin in this context could lead to new breakthroughs in medicine. In the second part of the session, Prof. Wendy Bean showed us just how these spin effects might translate into macroscopic outcomes. She reiterated Prof. Aiello's focus on how spin dynamics can be changed by weak magnetic fields of less than 1 millitesla, either promoting a singlet state where your spins are in opposite direction, which could lead to recombination of the radical pair, or alternately promoting a triplet state where the spins are in the same direction, leading to a reactive free radical with downstream biological effects. This is important in the context of a specific type of radical species, reactive oxygen, which play important signaling roles. And of these, Prof. Bean focused specifically on hydrogen peroxide and superoxide. Ross worked on a threshold effect, the hormesis that Alistair Nunn talks about, and both too little or too much can trigger disease. So Prof. Bean showed a variety of really interesting results, which I don't have space to include. The summary of which is very simply that weak magnetic fields have an effect on stem cell mediation of tissue growth in vivo in planaria. And planaria have powerful regenerative properties. And this is through the manner in which they modulate ROS, because ROS play an important role in stem cell proliferation, differentiation, and the process of regeneration. The results show more specifically that weak magnetic fields modulate blastema formation in wounded planaria, regulate injury-induced ROS accumulation, and regulate stem cell division and subsequent differentiation through downstream activation of heat shock proteins. What is particularly interesting is that the strength of the magnetic field, either 200 or 500 microtesla, which is not a huge difference, can give rise to opposite effects, either promoting or inhibiting these effects. Prof. Bean also showed that the reactive oxygen species that is responsible for this effect is not the hydrogen peroxide, but the superoxide, which is consistent with a lot of the radical pair literature. To conclude in her own words, ROS signaling promotes heat shock protein expression, stem cell mitosis, and new tissue growth. Weak magnetic fields affect growth by changing superoxide level. Weak magnetic field effects vary by field strength, both inhibition and activation. And weak magnetic fields may be a potential tool to manipulate stem cell activity, cancer, and or regeneration therapies. The fourth session of the series looked at mitochondria, bioenergetics, information, and electric fields, and the implications for repair and regeneration. Prof. Mike Levin, like Prof. Bean, is interested in the regenerative properties of planaria, but instead of magnetic fields, his research focuses on electric fields. Where are these electric fields located precisely? Well, unlike the Earth's magnetic field, these are endogenous electric fields or membrane potentials, which are the result of the difference of charge ions across membranes. In particular, he is interested in how bioelectricity can give us insights into how cell-level physiology scales up into anatomy and cognition. And what is the hardware that has evolved to allow for this scaling? Well, if genes only specify how to make proteins, how are complex morphological structures built from scratch or corrected when they are perturbed? And how might this be viewed as a type of collective intelligence? So neuroscience offers some tools with which to approach this question. Nerves have ion channels that help modulate their membrane potential and can propagate this potential to neighboring cells. That's their physiological hardware. And this underlies the cognitive goal-oriented activity of the brain. In a similar manner, the hardware that underlies morphological development is also ion channels and gap junctions, which mediate bioelectric signals that result in morphological outcomes. Prof. Levin then went on to discuss the hardware of morphology in more detail, particularly the V-ATPase complex, which creates a pH proton gradient, but also a voltage gradient. V-ATPase is implicated in regeneration across animals and plants. It also plays a role in symmetry in biological systems and drives the development of left-right patterning. This is because the creation of an unequal proton gradient controlled by the particular positioning of the proton pumps gives rise to this patterning. This is an extreme simplification of the details that he gave in his talk, which would take too long to summarize here, but it does give some idea of how the V-ATPase hardware translates into this anatomical scale morphological outcome. So to summarize in Mike's own words, all intelligence is collective intelligence, and biology uses this multi-scale competency architecture of nested problem solvers in diverse problem spaces, such as the physiological, the anatomical, and even the cognitive. And the thing that controls this is bioelectricity, which acts as a kind of cognitive glue, which evolution used to turn single-cell physiological energetic control loops into circuits that navigate collective decisions in morphospace, as well as potentially behavioral space. And the V-ATPase ion pump is a versatile piece of molecular hardware that is highly conserved in these roles, specifically scaling up from single cell level information to body-wide pattern. And learning to exploit this interface, we can improve control over growth and form. In the second half of this session, Prof. Wayne Frush took us through the finer details of this hardware, the ATPases. And ATPases play an integral role in mitochondrial ATP production. ATP synthase, for example, uses a pH gradient and membrane potential, which is both the difference in the number of protons as well as in the electrical charge, as an energy source for ATP synthesis. The protons move through a channel, which drives rotation of a drive shaft to force ATP production. And proton gradients and membrane potentials are very important in the context of mitochondrial electron transport chains, in which redox processes are coupled to proton transfer across membranes. Prof. Frush focused to some extent on the individual components of this V-ATPase hardware. For example, the C-ring subunit, which in different forms across species relates to the number of protons necessary to make a certain amount of ATP. For example, the C10 ring in E. coli would need 10 protons to make 3 ATP. I don't have time to go into the details of his investigation of C-ring proton dynamics. I'll only highlight the fact that the C-ring structures differ in different contexts, even though input and output channels of these ATPases are highly conserved. Frush then went on to discuss how these ATP synthases are regulated. In particular, the V-type ATPase has a particularly novel and interesting way in which it regulates how it works. It's highly regulated, being in touch with the metabolism of the cell. In the presence of glucose, there's glycolysis and the production of ATP. When glucose is low, the V1 dissociates and disables the pump. Again, this is a very small part of the many intricate details that Wayne presented, but it does give some idea of how the hardware that gives rise to Prof. Levin's morphological intelligence is very much coupled to metabolic concerns. In the final session, which was focused very explicitly on mitochondria and fields and implications for space travel, we had Prof. Afshin Beheshti present copious amounts of data that testify to the fact that mitochondria are affected by changes in the environment that result from space travel, in particular microgravity conditions and space radiation. I can't possibly cover all of the results that he presented, but will attempt to give an overview of some of the salient points, one of which is that space health might act as a sort of laboratory for medical research, where conditions that might take a long time to develop on Earth are accelerated in space travel. And some of these health risks include renal and kidney issues, liver issues, ocular issues, the central nervous system can be affected, circadian rhythms are affected, there's muscle degeneration, bone loss, even cancer risk. So a comprehensive cross-section of health problems. Prof. Beheshti presented a lot of evidence, which would take too long to go into here, but in summary, it can be said that space conditions have profound effects on mitochondria, analyzed on the level of genes and proteins, as well as metabolic and systemic processes. A very large amount of data was looked at by experts in many different fields, and when they compared notes, it turned out that mitochondria always ended up at the top of the list. The details are too many to list, but the data showed that across all cell types, there were universal mitochondrial changes. And future research aims to look at how this compares across species. He then moved on to talk about microRNAs and how these might relate to mitochondrial changes in spaceflight, and more excitingly, act to mitigate this. A single microRNA has been estimated to regulate up to 500 mRNAs. MicroRNAs are about 22 nucleotides, and due to the size and the stability of microRNAs, they can float somewhat freely in the blood. MicroRNAs are involved in all aspects of diseases, and they are found in everything that is living and are highly conserved across species. What is exciting about microRNAs in the context of space health is that they can be used as a biomarker, and even better, as a way to treat damage incurred by space travel. And again, this is a vast simplification of all the detailed studies that he presented. But in summary, there seems to be rigorous confirmation that space conditions have profound effects on mitochondrial function across all organs. And one way to measure and mitigate this is by targeting a specific microRNA signature. This has relevance not only for space exploration, but also for the way in which accelerated health decline from space travel may act as a model for improving medical practice on Earth, and in particular for mitochondria under stressful conditions that we have begun to examine in this series. Before I hand over back to Jeffrey, just one announcement on a completely different topic. Next year marks 100 years since the discovery of biophotons. In 1923, Alexander Gurwitch detected ultra-weak photonic emissions from onion roots, which he named mitogenetic radiation, but which came to be known as biophotons. He was convinced that they played a role in embryogenesis and the division and proliferation of cells. And he conceptualized this idea of a morphogenetic field, which unfortunately fell out of the spotlight with the discovery of genetics. And 100 years later, his ideas are finding a new audience as it becomes clearer that genes are important, but fields such as electric and magnetic and gravitational fields might also play some role in shaping living systems. And while a number of follow-up experiments were attempted, it proved really hard to reprove his original results until the invention of more sensitive photon detectors. And what this is leading up to is that to celebrate the centenary of the Gurwich's discovery of biophotons, the Gaia Foundation is launching a competition to reward the best or most novel, imaginative replication of Gurwich's original experiment. It's open to any researcher worldwide. The deadline for entries is the 26th of September next year, and the winner will be awarded the Gaia Foundation Onion Prize, comprising the opportunity to present the work at a Gaia Foundation online meeting and a cash prize of $5,000 US. The prize will be launched in December. If you'd like more details, check the website or the issue of the newsletter, which will be released next month. Thank you very much.